first FRQ. I've got some practice problems that we can do as well if you guys don't have specific questions. A. For the first FRQ? Like the first one for today or the first one period? Confused about A for the first FRQ. The pattern of inheritance? Okay. So for mitochondrial inheritance, it's always coming from the mom. That's just something that you have to remember. Let's see here. So for fertilization, you've got egg and sperm. And both the egg and the sperm have mitochondria, but the mitochondria in the sperm doesn't make it into the egg. So when sperm fertilizes egg, really the only thing that's dumped into the egg is the nucleus. And you've got the nucleus of the egg. Those two will fuse and you'll get that diploid organism. But for this particular problem, it's not genetic material inside the nucleus that's mutated. It's genetic material inside the mitochondrion that's mutated. So mitochondrial inheritance, that's the way it works. And it's not, let's see, what's the other part of it? Oh, individuals are typically not heterozygous with respect to mitochondrial genes because there's no backup copy inside the mitochondrial DNA. It's not diploid. So if you zoom in on the mitochondrion, you've got the outer membrane, you've got the inner membrane. This is like a gel space called the matrix, and it's within the matrix that you find circular DNA. So the DNA inside the mitochondrion is very similar to the circular DNA that we find inside of a prokaryotic cell. But the point is that there's no backup copy of anything. So, if this is the sequence of DNA where the mutation is, that results in the misfolded protein. I think they say it's a electron transport chain protein, right? Yeah, it helps accept electrons from NADH. So it's probably like, remember, the electron transport chain is made up of a complex of proteins. And so it's probably a mutation in this protein right here. This is the one that's going to be accepting electrons from NADH and then dumping it further down into the chain. But there's no backup copy. So if there was a backup copy, it would look something like this. I need to make these smaller. If there was a backup copy, there would be like 2 loops, and each loop would have all the same genes, but this loop might have a slightly different spelling at this location. If that was the case, then individuals could be heterozygous. They could have a mutated version and then they could have a non-mutated version. But that's not how it works. They've just got this little loop right here of DNA. Does that help? Yeah. All right, let me know if that didn't help. But in the meantime, I'm just going to jump through some of these cladogram problems. These are similar problems that you'll see on your assessment on Monday, your open note, open resource assessment on Monday. I've just pulled these off of old FRQs, publicly available FRQs. All right, so for this first one, it says mammal milk contains antibodies that are produced by the mom's immune system and passed to offspring during feeding. The milk also contains a sugar and many proteins, as indicated in the table. And they're asking us to use these characteristics in order to make a hypothesis about the evolutionary relationships. So we're not looking at the DNA sequences, we're not looking at the amino acid sequences, we're just looking at whether or not they produce a particular sugar or a particular protein in the milk. All right, so see if you can place these animals on the cladogram correctly. Notice that we've got a polytomy, so there should be 3 animals here that look exactly the same, and we can't further resolve those relationships. We would need more information to do that. And so I see 2, let's see. So I've got a cow right here that produces all of them. I've got a horse right here that produces all of them. I've got a pig right here that produces all of them. So there's our polytomy. I'll put a pig here, a horse here, and a cow here, right? So this is saying that they all descended from a common ancestor. This is the ancestral population that they all shared at one point in time. But we don't know if a pig is more closely related to a horse or if a horse is more closely related to a cow. So we don't know if it should be like this. Maybe you think it really should look like this. You can make that argument if you want, but based off of this evidence, we can't make that argument. We have to create the polytomy. And then obviously, just through elimination, we can put our cat and our human down here. And again, the way that you order pig, horse, cow doesn't matter. The way that you order cat, human doesn't matter, as long as you're saying that cats and humans share a recent common ancestor, and pig, horse, cow share a recent common ancestor. And then, of course, we all share a recent common ancestor, even further back in time, right here. The dark blue one. I'm not going to go into the immune response because this was from the 2014 exam, and that's been taken out of the curriculum. Yeah, so most of this just goes into the immune system. I just wanted to get the cladogram practice. Okay, here's another cladogram practice, and in this one, I think this is I can't remember. It's from a recent one, but they ask you to choose the cladogram that is, that was most likely constructed from this data. And so here we're just looking at whether or not specific DNA sequences are present or absent. And I think when you get information like this, the best thing to do is just to look at the outgroup. The outgroup is kind of like the control, because the outgroup by definition is supposed to be the group that is least related to all of the other lineages. So, for example, this is saying, this top cladogram is saying that a camel is more closely related to a cow than it is to a whale. In this cladogram down here, it's saying that a pig is more closely related to a cow than it is to a camel. It's also saying that a whale and a hippo are more closely related to each other than they are to any other organism. And so based off of this, we would say that a hippo is more closely related to a whale than a hippo is to a cow. And this one up here, we're saying that a hippo is more closely related to a cow than it is to a whale, right? So these two cladograms are saying some dramatically different things. And so let's see what the data shows. So in the top one, the whale is the outgroup. So the whale should look everything should look much more closely related to each other than the whale, according to the top cladogram. But if you look at this, you can see, let me make this, that when you compare the whale and the hippo, there's 1 similarity, 2 similarities, 3, 4, 5, 6, 7, 8, 9. So 9 out of the 13 locations that we're looking at, there are similarities between the whale and the hippo. So that doesn't really make sense to put the whale as the outgroup and put the hippo, the top one is saying that the hippo is more closely related to the deer and the cow. So there were 9 out of 13 similarities between the hippo and the whale. Let's look at how many similarities there are between the hippo and the cow, just for example. So I'm looking at hippo and now cow. So that's going to be 1. Not looking good so far. 2, 3, 4, 5, 6. But there were 9 similarities compared to the hippo and the whale. So this is looking good. And then let's look at the camel. The bottom one has the camel as the outgroup. So the camel, none of the sequences that we're analyzing are present. None of the sequences are present. So it looks like the bottom one is more supported by the data than the top one. And if we don't need to do this because we would be burning a lot of time on the clock, but if we wanted to, we can go and count how many differences exist between the camel and the pig, and the camel and the hippo, and the camel and the whale. And what we would find if we did that is that what we should find is that the number of differences between this group compared to the camel, it's a big number, and it's all going to be relatively the same. So like, let's just say if the cow has 7 differences between the cow and the camel, the deer should have approximately the same. The whale should have approximately the same. The hippo should have approximately the same. They should all be approximately the same different compared to the camel. Because remember, the fact that the peccary is right next to the camel on the bottom one is just arbitrary. I can take this node right here and pinch it and flip it, and I can make the cow be right next to the camel. So the fact that the cow is all the way to the right doesn't mean that the cow is the most different from the camel. Right? All of these lineages in light blue should have approximately the same differences compared to the outgroup. And so I just made up the number. If the difference is approximately 7, they should all be approximately 7 different differences compared to the camel. And the same would be true if the top cladogram were accurate. Like, whatever the number of differences exist between the whale and the cow should be approximately the same no matter which lineage we analyze. But we already saw that that's not true for the hippo. The hippo and the whale, there were only, 4 differences? And when we compare, let's see, let's do there should be more differences between deer cow. Let's see. Well, no, let's just see. No, I'm just curious. Well, we can't even use the top cladogram to analyze the data because the top cladogram isn't built based off of the data. So never mind. Okay. Let me know if you have questions about any of those. Let's move on to this one where, again, we're looking at amino acid sequence differences, and they've given us a template, and our job is just to fill in the genus and species names into the correct slots. So the first thing you should notice is that we should find two genus and species names that have very few differences from each other for this slot. And we should also find two that also have very few differences from each other. And so if I look up here, well, I already see here's a pair where there's very few differences, Africanus and Ferus. And then here's another pair where there's very few differences between them, Gallus and Forsteri. Now, does it matter where I put which one? If I want to put, let's see, if I want to put Africanus and Ferus, making your own cladogram from the chart. Like this chart right here, or this chart right here? The whale hippo one or the milk one? So for this one, I put Africanus here and I put Ferus here, and that means I'm going to have to put, what is it, Gallus and Forsteri here. Could I have flip-flopped them? Can I put Gallus and Forsteri on the right side and Africanus and Ferus on the left side? Yes, it doesn't matter. So all that matters is that you put the two species that are very closely related to each other coming from a recent common ancestor, which is what these nodes represent. And then of course the outgroup is going to be the group that is most different from everyone else, and that's going to be I believe it's poly, right? Poly is the only option. And notice that everyone is approximately the same number of differences between poly and whatever species we're analyzing. They're all approximately an average of 20 amino acid differences. Yeah, so for the whale hippo one, I don't think they would ever give you something where like they would ask you to build that cladogram from scratch. In this case, they just give you basically two hypotheses or two arguments, and they ask you to choose which argument is best supported by the data. And so if they give you something like that, my advice would be, I say they, but it's going to be us, because you're not actually going to be evaluated on cladograms on the test in May. We just need to get another summative in, at least one other summative in. But if I give you something like this, I would find a lineage. You can either look at an outgroup and then compare that outgroup to the in-group, or I would just find a lineage that shares a recent common ancestor and see if it makes sense. So, like, let's look at the hippo. So the hippo, according to this cladogram on top, the hippo should have a lot of similarities to the deer and the cow. And it should have more similarities to the deer and the cow than to any other group, right? Because they share a recent common ancestor according to the top cladogram. It shouldn't necessarily be more closely related to the deer than it is to the cow, because I could just as easily put the cow here on the left and the deer on the right. So I'm not making any claims about whether it's more closely related to the deer or to the cow. I'm just saying that it should be more closely related to the deer and the cow than to any other group. So let's see if that makes sense from the data. So if I look at the hippo, and here is my deer and here's my cow. So I'll do hippo to deer first. and I'll just count the similarities. No. Okay, so that's 1. That's a no. That's a no. 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 We can't really use that because it's undetermined for the deer. 2, 3, 4, 5. Right, so there are 5 similarities between the hippo and the deer. There should also be, according to the top cladogram, approximately 5 similarities when you compare the hippo to the cow. So let's see if that's what the data shows. All right, so that's nope. 1. 2, 3, 4, 5, 6. Okay, so far so good, right? It's got 5 differences, or 5 similarities to deer and 5 similarities to cow. We should also see, well, we can't really compare deer and cow in this case because both cladograms say deer and cow are equally different from each other. So now let's look at what the hippo's relationship is to the whale, because according to the top cladogram, the hippo should have a lot more differences when you compare the hippo to the whale than to the deer and the cow. So the number of similarities, in other words, should be much, 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 much lower. There should be fewer similarities according to the top cladogram when you compare the hippo to the whale. So let's do that. All right. No, no. There's 4 yeses right here. There's 3 yeses right here. And there's 2 yeses right here. So 4, 5, 6, 7, 8, 9. So there's 9 similarities, but there should be very little, right? If this cladogram is saying that the hippo is most closely related to the deer and the cow because there's 5 similarities, then how does it make sense that it has even more similarities to the whale, yet it's not closely related to the whale? Wait, so when there's only a difference of 1 similarity, then yes, you can consider them equally similar because it's the same logic over here. The numbers aren't going to always be exactly the same, but what we can say is that all of the organisms within this group should be approximately equally different to the outgroup, which in this case is Polylepis. And we see that rule holds true. It's not exactly 20, but they all look like they're like around 20 differences compared to the outgroup. Now, the thing about comparing the hippo to the deer and the cow is that the fact that we only see 5 and 6 differences between them, that makes sense also looking at the bottom cladogram. Like if you look at the bottom cladogram, this is saying that the hippo and the whale are very closely related because they descended from a recent common ancestor, so there's 9 similarities between them. And it still holds true over here to say that there's 5 and 6 similarities when you compare the hippo to the deer and the cow. In this one, on the top one, let me use a different color. The top one, so according to the top one, the camel should have more similarities to the hippo than the hippo does with the whale, because they share a recent common ancestor. So there should be more in common when you compare the hippo and the camel than when you compare the hippo to the whale, according to the top cladogram. So we already know that the hippo and the camel have 9 similarities. So if this top cladogram is true, then the camel needs to have more than 9 similarities in order for that argument to make sense. So let's compare the hippo and the camel. We need to see more than 9 in order for this to make sense, the top cladogram to make sense. Basically, everywhere there's a minus, there's a similarity, because the camel is all minus. So this is definitely not more than 9. This is only 4. Right, so this top cladogram just isn't making sense based off of this data. All right, let me see if I can find, I'm still looking at the chat. I'm just seeing if I can find more practice problems for us to work through. Okay, here's one, a Hardy-Weinberg one. All right, see if you can work through this one with me. Okay, it says for a particular genetic locus in a population, the frequency of the recessive allele, which we're going to use Q, is 0.4, and the frequency of the dominant allele, which is P, is 0.6. All right, so they make this easy for us because we don't have to do anything with like square roots or work with fractions. It just gives it to us straight up. So what is the frequency of each genotype? Big A is just P squared. Big A, little a is just 2PQ, and little a is just 2 squared. So that's going to be what, 0.16? .48, and what is that, 0.36? Whoops, sorry. I flip-flop my P's and Q's. 0.36, and then Q squared is 0.16. And that should all add up to equal 1 if I've done the math correctly. 0.36 plus 0.48 plus 0.16. Okay. You might get a problem where I actually give you the population size and I say it's 100 individuals or 1,000 individuals, and then I ask you what is the number of individuals who show the dominant phenotype. So assuming that the heterozygous looks the same as the homozygous dominant, then the number of organisms that show the dominant phenotype is just 360 plus 480, because you're taking this percentage and this percentage, you're multiplying it by the total number of organisms in the population, and then you're just going to add them together to get the number that show the dominant phenotype. How can the Hardy-Weinberg principle of equilibrium be used to determine whether or not the population is evolving? Right? So remember, the population is not evolving if the allele frequencies are stable over time. So if I come back many generations from now and I sample P and Q again and I get the same frequencies, well then that means that the population is in Hardy-Weinberg equilibrium, so it's not evolving. Another way we can use Hardy-Weinberg equilibrium is, so let's kind of like work on this from the back end. Let's say we see 160 individuals in a population of 1,000 who are homozygous recessive, and then I ask you to solve for P and Q, right? So just pretend that we haven't seen the problem above. Let's just work on it through this perspective. So 160 over 1,000 is just 0.160. So that equals Q squared. Well, if we want to estimate Q, we need to take the square root of that. Which should be 0.4, right? Whoop. Good, okay. Should be 0.4, so P equals 0.6, right? So far it's the same thing. But then we can use these numbers to estimate the expected values, right? So the expected values would be just the same as above. The expected P squared would be 0.36, which would be 360 individuals. The expected heterozygous would be 2PQ, which would be 480 individuals. And then obviously Q squared is what we actually measured, 160 individuals. Now, if the population is in Hardy-Weinberg equilibrium, then this is what we should see. But what if we actually go and genetically test the population and we see that instead of 360, we see 300 individuals who are homozygous dominant, and instead of 480, we see something like 520, and then we see 160. That doesn't make sense, right? No, it would have to be 540, yeah. So is the population in Hardy-Weinberg equilibrium if what we observe is a little bit different than what we expected? And that's where the chi-square test comes in, right? Because the null hypothesis is just the population is in Hardy-Weinberg equilibrium, and the only reason we're getting different observations versus what we expected is just randomness. Has nothing to do with violating Hardy-Weinberg equilibrium. That's what the null says. So we can do observed minus expected, square it, and then divide it by expected. Right? And we can do that for every single, I'll do that over here, every single category. I'll remind you of this on Monday, but remember that the degree of freedom for a Hardy-Weinberg equilibrium problem is 1, because there's 2 alleles. So 2 minus 1 is 1. All right, I'm going to work the O minus E squared divided by E out, and then we'll actually do our analysis. Alright, so I got 17.5 as my chi-square sum. This was equal to 10 this was equal to 7.5, and this is equal to 0 because 160 minus 160 is 0. We add it all up, and we get a chi-square sum of 17.5. Our critical value, and you guys will always have a chi-square table to reference. Let me pull up the one that you'll actually have on your test. I don't know if they have that posted under the course and exam description. No, hang on. Okay, here we go. So it was 17.5. In this case, the critical value is always going to be 3.84 because our degree of freedom is always going to be 1, assuming that the number of alleles that we're working with is 2. And I'll just go ahead and tell you it is. Like, you're not going to see Hardy-Weinberg on your test in May. I'm not going to give you a weird Hardy-Weinberg on your test on Monday. You're going to see something similar to this. So your degree of freedom is going to be 1. Your critical value is going to be 3.84. So it was 17.5 is the chi-square sum. You compare that to a critical value of 3.84. Remember, the way that I remember this is high equals by. So if the chi-square sum is higher than the critical value, you say by to the null hypothesis. If it's greater than or equal to. If the chi-square sum is less than the critical value, low is no. You do not reject the null. In this case, it is higher than the critical value. So we say by to the null. We would say that this population is experiencing evolutionary forces. So one or more of the Hardy-Weinberg assumptions is being violated, according to our data. The population may be experiencing natural selection, it may be experiencing sexual selection, it may be experiencing mutations that are altering allele frequencies, it may be experiencing gene flow, so organisms may be coming into or leaving the population, which can alter allele frequencies. And then the last thing is that it may be experiencing genetic drift. There's a picture of genetic drift that I think does a good job of just summarizing like what it is. So when I think of genetic drift, this is what I think of. So let's imagine you've got a population of beetles, and the green coloration is due to a homozygous recessive genotype. And so if you've got that brown coloration, you're either going to be big A, or you can be big A, little a. But as long as you have that dominant allele, you've got the brown coloration. And so in this example, because the population size is so small, any random event that happens, like this person, green is not disadvantageous because this person is looking to kill green beetles. This person is just walking along, not paying attention to what they're stepping on, and they just happen to step on these green beetles because they happen to be in the wrong place at the wrong time. which is going to cause that allele to dramatically drop in the population. So this is a huge random swing in the allele frequency that has nothing to do with natural or sexual selection. It just was randomness. And so when the population size is very small like this, imagine a scenario where you've got a massive population of beetles. You've got like 10,000 beetles, and 2 green ones get crushed because they were in the wrong place at the wrong time. That's not really going to do anything to the allele frequencies. That's just a drop in the bucket. But if your population size is really small, like 9 beetles, and 2 of them that are green get stepped on and get crushed, then suddenly the allele frequencies just swing wildly in one direction or the other. Let me pull up a simulation that I showed on a video when you guys were looking at the parrots getting picked off by the eagles. Give me one sec. All right, so here is a population where I think the population size is like 4,000. And I put the population in Hardy-Weinberg equilibrium, so you can see over time P and Q, it looks like it starts at around 0.6 and 0.4. Let me set the initial P and Q conditions. Let's just do, let's just make it 0.6 and 0.4. So over time, it fluctuates a little bit because sex is random. But for the most part, it stays around 0.6 and 0.4. Now, you suddenly drop the population size from 2,000 to 20. I'm gonna make the P and Q frequency 0.6 and 0.4. Notice that when I run the simulation, every time I run the simulation, the allele frequencies just go crazy. They do random, unpredictable movements every time I run the simulation. So this is what this is where genetic drift can be really bad. This population, every population is going to experience a little bit of genetic drift. Genetic drift just means random changes. But if the population size is large, that genetic drift won't have a significant impact on the frequency of P and Q. If the population size is small, genetic drift can cause the P and Q frequencies to go crazy, which is not a good thing. Like whenever P and Q frequencies randomly change, that can be bad for a few reasons. The first reason is that genetic drift can reduce genetic diversity, and it reduces genetic diversity most of the time when it does affect a small population, because typically genetic drift can lead to allele fixation, or that's just another way of saying one allele disappears and the other one is 100%. So notice that in a lot of these simulations, I get allele fixation. So that number one, it reduces genetic diversity, which like defeats the purpose of sex. Like the whole point of sexual reproduction is to shuffle those alleles and increase genetic diversity. But if everyone has the same allele, then there's no shuffling to get new combinations. You're just getting the same combinations every generation. The other bad thing about genetic drift is, what if green is actually advantageous. Like, what if green beetles have a higher fitness because they can better camouflage against predators? And these green beetles, again, just happen to be in the wrong place at the wrong time, and they were crushed. Then suddenly that advantageous allele is wiped out of the population, or it's dramatically reduced in the population, and that can make the population overall more susceptible to extinction if you're randomly just erasing good alleles, advantageous alleles from the population. Okay, let's see if I can find what else. What else do we have? The only other thing that I can think of is pre and post zygotic barriers are. This is what the bird video, when you guys were working on the lizards and how they occupy different vertical niches. And then you saw the bird video that talked about geographic and behavioral isolation and how that can be a catalyst for speciation. I'm trying to find a good... wait, I had a figure in that activity. Let me see if I can pull that up real quick. Something like... maybe it was this one. I think it was this one right here. Yeah. So just on Monday, just be familiar with what it actually means to be a member of a species. Like, what is the biological definition of a species? And the answer to that is that it's just a if you are a member of a certain species, that means you belong to a population of organisms that are capable of interbreeding, making fertile babies. So if I see two organisms and I want to know whether or not they're members of the same species, do they interbreed and can they make fertile babies? And if the answer to that is yes, then they're members of the same species. If the answer to that is no, then they are different species. And so these are just the sort of the two major mechanisms that keep members of different populations, where was I? You can think of these mechanisms as like a wedge. So if I've got, population A over here, and they're a part of one species, and I've got population B over here, and they're members of another species, these pre and post zygotic barriers are like little wedges, or look like a wall that prevents gene flow between those two populations. Gene flow is just another way of saying interbreeding, mating. So once you separate those gene pools into two distinct gene pools, the P and Q frequencies of all the different alleles for all the different genes can begin sort of traveling or changing down their own course. And so this population over here can experience their own P and Q changes. This population on the right can experience their own P and Q changes. And if those changes aren't being shared between those two populations, then they will continue to genetically diverge. And if enough time has passed, these mechanisms will sort of solidify between those populations, and that's really going to prevent those organisms from interbreeding. And so for the bird video and for the Anolis video, it was primarily behavioral and ecological mechanisms that led to the divergence of all those different species. The behavioral aspect came into play when they were talking about sexual selection and how they have the little colored dewlaps, and that allows, you know, the females to ensure that they're mating with males who are members of the same species. And then the ecological aspect came into play when you had, you know, different populations of lizards that evolved and developed adaptations to better exploit their own little niche that they occupied, whether they, you know, exploited the branches or the trunk or the, you know, the brush. So these mechanisms up top are just going to prevent members of different species from ever attempting to mate in the first place. But every now and then you have members of different species who come together and they mate. And if they are, we would still consider them members of separate species if the post-zygotic isolating mechanisms affected the offspring. And so that just means that if the offspring is infertile, then the parents are still members of different species. If the offspring is like sickly, like they typically, because let's say the genetic differences between the mom and the dad are so extreme, and there's so many developmental abnormalities in the offspring that they die before ever reaching sexual maturity. That's what's called hybrid inviability. And so again, mom and dad would still be considered members of different species. And then hybrid breakdown it's really the same thing as hybrid inviability. It just takes longer for that to manifest. It might not happen after the first hybrid. It might happen after the second or the third hybrid. But the point is that the babies are all genetic dead ends. If the baby is a genetic dead end, then the parents are still considered members of separate species. So I would just make sure that you're most familiar with that through the lens of the anolis lizards. And again, you're going to be able to reference this on your assessment. There's not going to be any curveballs or trick questions here, but it helps to know that going into this. You guys are not going to have to worry about, like, do I need to go back and remember how to be able to use this cladogram building software. Just focus on the concepts. Calculating the allele frequencies with a given number. Oh, yeah. That's probably one of the easier problems that you could get. If I tell you that big A equals 20 and big A equals 10 and little a equals 30, well, the thing that makes this so easy is that typically when you are given the phenotype of a population, you're given the dominant phenotype and you're given the recessive phenotype. And you know that the recessive phenotype equates to one and only one genotype. But for the dominant phenotype, you don't know how many of those people are big A, or how many of those people are big A, little a. Now you can guess, you can make an estimation by doing the whole Hardy-Weinberg number crunching, but if I actually tell you, then all you have to do is just count up the number of big A's and divide by the total number of alleles, and then count up all the little a's and then divide by the total number of alleles. So how many big A's are there? Well, if there's 20 people who are big A, that means there are 40 big A's from this group, and there are 10 big A's from this group, so there's 50 big A's. How many little a's are there? Well, there's 10 from this group, and there's 60 from this group, so there's 70 little a's. How many total alleles are there? 120. If I did my math right, yeah, 120. So what's the frequency of P? Well, that's just the frequency of big A. So that'd be 5 over 120, or 50 over 120, or 5 over 12. What's the frequency of Q? Well, that's just 70 over 120, or 7 over 12, whatever that comes out to be as a decimal. I think the activities that are going to be most helpful, if you look at your roadmap, it's all in one place. PKU is going to be helpful. The rock pocket mouse is going to be helpful. That's the making of the fittest. The Anolis from that Monday and Tuesday before spring break is going to be really helpful. The virus, don't worry about that on the assessment, like the HIV, how it infects, the CXCR, whatever, CXCR4 versus CCR5, that won't be on the summative. Cladograms, absolutely. The cladogram practice questions. And I don't know if you were here at the beginning, Kimberly, but this is going to be uploaded. I went through like 3 or 4 cladogram practice problems just by looking up AP Bio FRQs on cladograms. And you guys have done what I think at least 2, if not 3 cladogram problems on the practice FRQs that I've been posting. And that's it, right? So those are the big ones. The Hardy-Weinberg, making of the fittest with the rock pocket mice, Anolis. Maybe go back and if you want to redo the Hardy-Weinberg formative assessment, or if you want to go back and redo, there were like 20 Hardy-Weinberg practice problems, I think. Right here. No, right here. That came directly from AP Classroom. When we make the cladograms, we're not going to have more than... Oh, no. I'm not going to ask you to make a cladogram like by giving you 16 lizards. I'll ask you to make cladograms similar to the cladograms that they would ask on the AP Bio exam if you were to be tested on it this year, but you're not being tested on cladograms on the AP exam this year. So like most of the cladograms that you guys have seen that have come from AP Bio exams, it's been like five organisms, right? So it's not nearly as complicated as the Anolis one. And I would never ask you to make like a lizard cladogram out of 16. I think I asked you to like pick one of your subgroups and try to build a cladogram from it. But I think it was this one, right? Like I might give you 4 and ask you to make one like I did on this one. I think I had 3 in that group. I don't know if this is what you're asking, but you might have a polytomy. Like I might give you, like, here's just a generic example of when a polytomy would be necessary. You have organisms A, B, C, and D, and we're looking at, you know, traits 1, 2, 3, and 4, and A, B, C all have traits 1, 2, 3, and then D has trait 1, but not, you know, 2, not 3, but it has 4. So based off this information, the only thing that I can really say is maybe something like this, right? A, B, and C go right here, and then D goes right here. With the PKU assessment, yes, I thought I had updated it yesterday, but if you feel like that your grade is not what you thought it would be, I went back and gave you 100% credit if you corrected it. Like full credit back. I will be on again tomorrow from, what did I say, 9 to 10? Yeah, I think I said 9 to 10. But if you have any more questions throughout the day, just email me. That was on the FRQ yesterday. Yeah, I can do that. Can you re-explain the thing about switch? Oh, yeah, I know what you mean. Okay. So, with the polar bear switching thing, yeah. So let's say I have a cladogram that looks like this. Okay, and I have D, I'll put A, B, C, D, and E over here. Okay, so I'm going to draw this as like a river. So it's a river of time, okay? You've got some sort of ancestral population, like let's pretend we're at a bird's eye view, right? So we're looking down on all of these organisms in this population, right? And what we're seeing is the population splits like this, okay? And they go down their own way. They stop interbreeding with each other. And as a result, you get speciation. And this population on the left just kind of goes their own way, and they eventually get to, I'm running out of room, hang on. I know this isn't exactly what you're asking about, but I feel like this will help make sense about what the whole switching thing means. It's not letting me move it. Well, I'm just going to delete it. Okay. And then you have this population over here on the right, like here, like let's say this is present day. It's like a river of time, right? And this was off in the distant past, like 325,000 years ago, or whatever the answer was to that question. All right, this group over here, they're going to split again into something like that, and split one more time right here. And the same thing's going to happen over here, right? They're going to split one more time sometime in the distant past, and we're now everyone's at present day, right? And they're going to keep flowing through time. We're just seeing them at present day, right? Now we can go through and like label the ancestral population. Right, so here was the ancestral population for this group right here. Let's just call this group C and D, right? And here was the ancestral population for this group right here that would eventually split and lead to A and B. Now in my cladogram, it doesn't matter where I put A and B and C and D, as long as I put them branching off of the same ancestral population, right? So if I'm drawing this, and we're going to call this group over here E. Ooh. And the ancestral population is right here. That all of them share is right here, right? So if I wanted to turn this into a cladogram, I can do this. I can put A and B right here. I can put C and D right here, and then I'll link them together, and then I'll put E over here, right? So this population corresponds to this node. And I forgot to draw this in. Here would be the ancestral population that would split and lead to the C, D, and AB lineages. So that corresponds to this point right here. This corresponds to this point right here, that ancestral population. And this corresponds to that point right here. So that's what the cladogram is saying. And so in this case it doesn't, like, if I were to take this river, I don't know how to, if I were to like grab this chunk of my river and rotate it, like flip it, and so A and B were on the left and C and D were on the right, that doesn't change anything about the information. They still diverge at the same point in time. They still diverge from the same common ancestors. It doesn't matter how I choose to represent it in that aspect. So I can just as easily write a cladogram that says BA, doesn't matter, they're still linked to the same point. And I can put D here and C here, that doesn't matter because they're still linked. And I'm still linking A and B and D and C together, so that doesn't change anything. And then I'm still putting E out here, so that doesn't change anything. So the rule is that, and I can even pinch it and flip it at this node right here, right? So I can pinch it and put D and C on this side and A and B on this side, doesn't matter. Right? The information is not changing. I still have D and C branching off of an ancestral population, which they're doing right here. I still have A and B branching off of an ancestral population, which they're doing right here. And I'm still putting E, flipping E out here. Or, you can flip it and put E out here. Doesn't matter. You're still saying the same thing. And so the reason that's important to remember is that a lot of students think that, because D is so close to E, that means that D is more closely related to E than it is to B. Because look, B is all the way out here, like on the right. But that's not correct to say that. D is more closely related to B because they share a more recent common ancestor, right? This is the common ancestor they share. They did not split as far back in time compared to when D split from E. If you want to find out where D split from the E lineage, you have to go further back in time. And so because of that, D is more closely related to B than it is to E. Another way that you can think about it is, like, as far as why it's accurate to say that D is more closely related to B. So this right here, you're welcome. Whoops. This right here is time, right? It could be thousands or tens of thousands of years of time. Well, that is time that where genetically, uh, this is a gene pool that is shared between the C and B lineages. Like this population evolved together for thousands or 10,000s of years before they started to diverge genetically. So all of this right here is time that C and B evolved together, that they didn't have, like C did not evolve together with the E lineage for all of that time. So there is more time for genetic divergence between the C lineage and the E lineage, which means that they're less related to each other when you compare C to B. C to B is more related to each other than C is to E. Okay, I'll be on tomorrow from 9 to 10, and then I'll answer emails. I can even be on like Monday morning. We'll just play it by ear, but that's when your assessment will be. You guys have a good day.